This method can help answer key questions in the field of cancer pathology by quantifying biomarker proteins from routinely processed biopsy material. The main advantages of this technique in relation to conventional immunohistochemistry are that it is objective, has a wide dynamic range, and permits for the quantification of proteins in specific cell types.
The presence or absence of certain so-called biomarker proteins, or more particularly the relative abundance of those proteins in cancer biopsy specimens, can be very useful in making a specific pathological diagnosis of specific kinds of cancers. But it can also be useful in predicting the response to cancer treatments. So that's why this technique is relevant both to the diagnosis and the treatment of patients with cancer. This protocol is mainly about how to use protein quantification in immortalized cell lines to validate the quantitative nature of an immunofluorescence-based assay. But it's important to note that immunofluorescence can easily be incorporated into a multiplexed approach and applied to primary biopsy samples.
Assisting in demonstrating this procedure will be Boudreau, a technician, and Shakeel Virk, the Director of Operations, both from the Queen's Laboratory for Molecular Pathology.
To begin, centrifuge the previously harvested cells in a 50 milliliter conical tube at 225 Gs for 5 minutes. Decant the supernatant and resuspend the cell pellet in 10 milliliters of PBS. Then centrifuge the resuspended cells at 225 Gs for 5 minutes. Suspend the cell pellet with 10 milliliters of 10% NBF. Then incubate the cells at room temperature on a rock.